everybody. Thanks for joining my oral presentation today. My name is Dr. Colin Hawco. Today I'm going to talk about individual variability in brain activity and how it relates to cognitive performance. The basic premise of my research is that we are not all the same. And I think this is something that we would all agree is obvious, but also that we forget about a lot in cognitive neuroscience. The majority of our research relies upon sort of looking at group aggregate averages. We take 20, 30, 50, 100, 1,000 people, we put them all together and say what on average does their brains look like. But what does an average really mean when no person might actually really fit that average template very well? I think honestly, while averaging approaches have their place and continue to have their place for years to come, we're approaching the limit of what we can learn from our current group aggregate approaches. And if we really want to move forward with our understanding of human brain function, we have to move beyond the group average and start looking at the range of variability we see across individuals. Particularly for those like me who want to apply this sort of information to clinical contexts. So the way I'm going to handle this is to use clustering, which is basically a way to put similar parts of the data, similar participants together and group them together and say these people look similar, these people look similar, these people look similar. So maybe they kind of become little subgroups. We're going to use the human connectome task in order to interrogate this issue more deeply. We pull data from the human connectome HCP 900 release, which includes 900 participants. And the HCP data set is pleasant in that it includes 6 task modalities and different cognitive fMRI tasks: emotion, gambling, language, relational, social, and working memory. So we took the HCP 8mm smoothed surface data, which they provided us for T-maps for all the tasks. So we now have individual activation maps. We can now take each person's individual T-map and extract it as basically a vector that we stack all these participants together, and we can pass it into a hierarchical clustering algorithm. We use Ward's method because it's what we found to be highly effective with high-dimensional neuroimaging data. We're going to examine data from solutions from k equals 2 to k equals 10 clusters, but for the most part, we're going to focus on the k equals 4 solution as the most representative solution. This data has been uploaded as a preprint to bioRxiv, so you can examine that for further information and details. And of course, I'm happy to answer questions offline. If you go to bioRxiv and you search my last name, Hawco, it should be the first paper to come up. It's called Moving Beyond the Mean: Subgroups and Dimensions of Brain Activity. So when we look at our initial clustering solution for the k equals 4, we find a very characteristic pattern in which we see at the top here we have a sort of a deactivating group, then we have sort of 2 intermediate groups at the bottom, a kind of a group showing a strong way towards positive activation. And this is true across all 6 tasks. We have a range of variability which goes predominantly from kind of a most negative deactivating group down towards a most positive activating group. And if you look at the bottom row, this is the full sample group average, what we would typically look at in studies. But I think it's pretty clear if you look at, for example, the relational task, the cluster on the top, cluster 1, and cluster 4 on the bottom. In fact, none of the clusters really do a close fit to that group aggregate map. So the group map might suggest that people are engaging in both task positive or task negative systems, when predominantly there tends to be either one or the other. So in this way, group averages can actually miss some important variability across the data and even potentially mislead us. Our primary analysis of interest here is to look at cognitive differences across clusters. So to deal with this issue, to assess differences in cognition across clusters, we did a series of one-way ANOVAs. We took 12 tests for the human connectome projects. We had at one point tried to do a PCA to reduce this to sort of a G factor or our average cognition value, but we didn't get a very good solution. So we decided it was better to treat each test as a separate data point. And so here we have the emotional task, for example. The rows are each of the 12 cognitive scores that we have for out-of-scanner cognitive abilities. And the columns are different cluster solutions from k equals 2 to k equals 10. And the cells that are in color, the ones that were significant after FDR correction, find differences in cognitive abilities across clusters. So across all tasks, we found a variety of differences between members of clusters and their cognitive abilities. But I'd like to highlight that the biggest differences we see are in the language, the relational, and the working memory tasks. And these are of the 6 tasks, the ones I think of as the most traditionally cognitive tasks. They're really the ones that load onto what we think of as normal cognitive abilities, sort of neurocognition. And those task fMRI results tended to map best onto these sort of out-of-scanner cognitive abilities, particularly things like Raven's matrix and reading, which are more related to sort of generalized cognition. So in order to zoom in on this, we're going to look at Raven's progressive matrices so we can really see which patterns of brain activation relate to which sort of better or worse or et cetera cognitive abilities. Raven's matrix is the closest thing we have to a sort of generalized cognitive test. It's often considered sort of a surrogate for fluid or verbal intelligence. So that if we look at particularly in this first, the language and working memory tasks, we see the cluster that performs the worst is the one that has the least activation. So they kind of have less activation in brain-driven tasks. versus the relational and emotional tasks, it looks like these sort of deactivated weighting clusters are the ones who are performing the worst. So in the case of the relational emotional task, this deactivating pattern is not good. In the case of language and working memories, the people who just aren't engaged as much who seem to be the ones who have the least cognitive ability to scatter. Now, one of the core goals of the study was to look at reliability of these cluster solutions. And I'm going to apologize in advance because I'm going to go through this very quickly. It's a bit complicated, so I would refer again anybody for more details to contact me offline or check the preprint. But basically what we did is we did a bootstrapping approach. We did 1,000 permutations where we took 75% of the sample and we reran the clustering solution. And this gives us a graph that looks like this. This is our cluster probability matrix. It says if two participants were in the same bootstrap, what's the probability they were also in the same cluster? This is a participant by participant matrix, and so the probability is from 0 to 1. And if we had perfect clustering, this matrix would look like a bunch of white squares, 4 white squares. This is the working memory K equals 4 example. But it doesn't. We have this sort of gradiated, kind of blurry cluster effect. So it looks like there's some sort of pseudo clusters, but people can actually jump back and forth between adjacent clusters quite a bit at different iterations of the solution. This looks to us almost more like a sort of a pseudo spectrum. So we really want to dig into this a bit deeper. So we can take this matrix in order to pull the salient piece of information out of it. We can form a principal components analysis. Here we can generate components. So each person gets now 1 component square per component. So this first component seems to represent something about the diagonal. The second component, I think, is something about how sort of bulgy that part of the graph is to some extent, up to a point. And now that we have everybody with just 2 scores instead of 822 correlations, we can plot a participant on the graph like this. So every circle here represents 1 participant. The colors are the original cluster solution, how they group together. So we see there is a sort of coherent grouping here. And we can add a third component, and now we're explaining more than 90% of the variance in this graph, and that folds the circle, but everybody still fits along the surface of this circle. And this to us represents a kind of a lower dimensional similarity sort of manifold space. It's the range of variables that people can have relative to each other. The bootstrap in this case is kind of a data-driven feature selection stage. And what we get out of it in this sort of what I think of as a taco plot is a measure of how similar or dissimilar people are in their patterns of activation of the task, based on the most salient features of the task activation patterns. Across our 6 tests, we get 2 characteristic shapes. We either get a folded circle shape like a tortilla or taco, or we can get these S-shaped kind of snaky shapes. For details into why we think those 2 shapes emerge, you can refer to the paper. But for our purposes here, we're interested in the relation to cognition. So what we did is we took each of these 3 component scores. Everybody has the component score in each of these 3 components. And we then related those back under cognitive abilities. And we find, again, there's loading heavily on the language relational working memory task, where this new sort of lower dimensional participant similarity space that kind of judges how similar is your brain pattern across different tasks now loads onto cognition in a way that's not just based on sort of a linear relationship between, oh, I took this one spot in the brain I kind of measured as we do in most common regression analysis. So this may be a new and interesting way for us to explore cognitive relationships across different patterns of brain activation without necessarily doing linear kind of regression type approaches for brain activation and cognition. So that's it. In conclusion, activation patterns are highly variable. This may not come as a shock to anybody, and I think most of us know this, but I also think we don't take this to heart in our day-to-day lives. The group map misses a lot of important individual variability. We need to move beyond that group map to really capture all this. This behavioral variability we see, or sorry, this task activation variability is behaviorally relevant. It's important. What we see in these cluster bootstraps and elsewhere might not map cleanly onto just linear relationships. If you look at these different 3 patterns, they might not have that. Well, that part of that brain is now linear maps on cognition between this group and that group. Maybe this group that has a different pattern that has a different linear mapping than this group if we think about how cognition loads on brain activation. And the long and the short of it is, as much as we might wish that we were, we can't all be John Malkovich. There's only one John Malkovich. And if we treat everybody as if they were John Malkovich, we lose a lot of important information about brain function. Task activation now seems to vary mostly along this sort of positive and negative dimensional axis. I don't think it's very clear to us why that is. It could be attentional effects. We examine the idea of hemodynamics, and I don't think that provides an answer. My preferred explanation is the sort of behavioral strategy effect that how people do this task in the scanner is what drives their patterns of brain activation more than anything else. And this cluster bootstrap reveals this sort of similarity manifold space that may be a new and exciting way to think about variability in brain activation and how that maps onto variations in behavior and cognitive abilities. That's all. Thank you all for your attention. Thank my colleagues and co-authors, and if anybody has questions, I'm happy to answer them now or by email.
Hello. Firstly, thank you very much for the invitation to present this work here at OHBM. My name is Ana Luisa, and I will next talk about the study on segregation of functional territories in individual brains that I've been developing together with my supervisor, Bertrand Quirion. So this work is in close relation to the individual brain charting project, a 10-year-long project which is still ongoing in the lab, and it pertains to the development of an open access neuroimaging data set, mostly task fMRI data acquired at higher spatial resolution, concretely 1.5 millimeters. And one unique aspect is its strong emphasis in a task-wise dimension, meaning that data is collected from many tasks, most of them reproduced from previous studies, in the same people, at the same place. So this facility offers us the appropriate setting to better investigate approaches in individual functional atlasing. So because we are gathering data from multiple tasks, commonalities in their functional representations can be associated with behavioral responses shared by them. I'd like also to stress that here we are interested to map these common representations in terms of the functional segregation of specialized brain regions and link them to mental functions that are putatively involved in these behavioral responses. So we are here also minimizing sources of intersubject and intersite variability across tasks because data has been acquired in a fixed cohort and environment. But we also know that the standard group level analysis can hinder statistical significance and finer demarcations of brain regions. So that's why we are here exploring the feasibility on performing individual brain modeling, profiting from the benefit that the data has been acquired at higher spatial resolution. So we are leveraging the first release of the IBC dataset, which is composed of 12 tasks, covering already a wide variety of psychological domain, ranging from perception to higher order cognition. And for more information, you can consult the data descriptor paper of this release that has been already published in a scientific data journal. So then we employed mass univariate GLM analysis to compute the most relevant volume-based contrast maps, and the corresponding threshold, the Z maps, are already available and properly annotated in NeuroVault. And these derivatives constitute the basis of our cognitive mapping approach. So I will next describe our first study on dictionary of cognitive components. So here we performed the decomposition of the 51 main contrasts with the dictionary learning into 20 components per subject. And these individual topographies provide a low dimensional representation of the contrasts and retain the effects of interest that are shared by them. So in terms of labeling, each component gets the name of the active condition from the contrast with the highest loading, because this condition is the one that has contributed the most to explain the variability of the signal of that component. So here we display the individual topographic maps, and we can clearly see that components are consistently mapped across subjects. And this is quantitatively confirmed by the median map presented in a red frame. And it represents at every voxel the component that is at least present in half of the subjects. So to gain some insights about the commonalities underlying the contrasts, we try to reconstruct contrasts of one task from contrasts of the remaining tasks. So we employed a leave 3 out cross-validation experiment to learn the shared representations from contrasts of 11 tasks and then predict all contrasts that were left out for validation. So we show here the maximum accuracy prediction scores obtained at every voxel, and this highlights the amount of all brain functional activity that can be predicted, and somehow this tells us how much these tasks share in common. And with a few exceptions, we see that most of the brain regions are covered by the predicted functional signatures. So another way to look at these results is in terms of the proportion of the voxels with the high positive R square. So here we can see in this plot the size of the brain regions that are functionally characterized by each task. But one interesting aspect to point out is the fact that if we permute subjects between train and test, this proportion decreases in all tasks, showing that topographies are indeed driven by subject-specific variability. So we can conclude that these findings favorably advocate toward individual brain modeling approaches. So I'd like to finalize by showcasing an application on functional mapping that benefits from knowledge accumulation of contrast maps. So we have selected a set of regions of interest previously reported to be engaged in language mechanisms and draw their cognitive profile based on IBC language-related contrasts. So I'd like to emphasize here that these regions of interest were made subject-specific by projecting their original masks into an individual template. And this was achieved by performing dual regression in the remaining contrasts, so not the ones that are presented, that were selected per subject. So for each region of interest, we plotted the average Z scores obtained at every voxel. And this shows us how these regions of interest are functionally characterized by the selected contrast. So to assess the statistical significance of these differences, we tested whether the voxels could be reliably classified as belonging to one of these regions of interest. So we computed the prediction scores within pairs of regions of interest using a linear support vector classifier, whose results are displayed in the upper triangle of the diagram, and also the corresponding chance level with the dummy classifier that you can see in the lower triangle of the diagram. And overall, we can see that the prediction scores outperform chance level. So in conclusion, we show that functional atlasing using a large data set in a task dimension support the investigation of common functional profiles between tasks that can be linked to shared behavioral responses and subsequently to mental functions. And also that individual brain modeling using data with higher spatial resolution successfully performs generalizations across subjects at the same time that elicits variability between them. So I'd like to announce that the second and third releases of the IBC dataset are already out. Source data is available in OpenNeuro, derivatives in NeuroVault, the documentation on the IBC website. And this research has been funded by the Human Brain Project. I'd like to thank to the IBC volunteers for their commitment to be scanned during such a long period of time, but also to all collaborators from INRIA and Neurospin in Paris, namely UniAct and Unicog Lab, but also to all my colleagues from the Parietal team, and in particular to my supervisor, Bertrand Thirion, for the all support and main insights about this project. And I'll be available to discuss about this work in a virtual poster session. And thank you very much for your attention.
Hello, I'm Matthew, and today what I'm going to be talking about is some work that I did with Anxu Wang, my RA, at the Washington University in St. Louis. So the question that we're interested in is how do we relate the more loosely organized brain activity that we observe in resting state with much more constricted patterns that we observe during task fMRI? And there are a couple of reasons why we're interested in this question. Of course, there's basic neuroscientific insight and potential clinical applications, if we can predict how changing network architecture changes brain activity during tasks. Potentially, we could try to predict how it will change brain activity in different contexts. But the question that we're really going to be looking at today is, can we use intrinsic brain activity to better estimate task effects? I'm doing this for two reasons. The first is this provides a good quantitative proof of concept of how we should relate task and spontaneous brain activity, as well as it also has real practical implications in terms of actually just performing task fMRI analyses. So the two main observations across task and resting state fMRI are that the resting state fMRI shows greater variability than task. That is, that there is a much wider space that the brain traverses in resting state than in task. We also observe preserved network architecture between rest and task, and that holds in terms of both the functional connectivity matrices and in terms of the static betas. For instance, Colin colleagues have shown that you can use resting state functional connectivity to predict the patterns of task-related brain activity. So the question that we're looking at is, how do we get from these more loosely organized spaces observed in spontaneous brain activity to the much more constricted spaces that we observe in task? And should our fMRI analyses reflect this relationship? The framework that we're going to be using is a dynamical systems framework. That is, we're interested in how the brain activity evolves in time. We're going to be having one model that we fit for the resting state brain activity, and then we're going to use a simple model where we just model task as an exogenous forcing onto the resting state activity. That is, we model task as having some exogenous stimulation that flows throughout the resting state brain dynamics and the endogenous architecture. The approach that we're going to be using to actually build these models is termed MINDI modeling. Mesoscale, because we're doing this in terms of parcels, we're going to actually be working with 419 parcels typically. Individualized, we fit these to each individual subject using the resting state fMRI. And neurodynamic, because these take the form of dynamical systems models. The main component of the model is a connectivity matrix that we fit to the resting state data. And so this is a signed and directed connectivity matrix, so it's estimating effective connectivity rather than just correlations. We also describe the way that different brain regions differ. We have two components that are region specific, namely a transfer function, which is a nonlinear function that describes how regions communicate to each other, and a decay parameter, which describes the time constant of different brain regions. So this is a phenomenological parameter, which describes how quickly or slowly regions integrate input over time, and shouldn't be confused with, say, the time constant of the neural membrane. We also perform hemodynamic models for each subject. So we're fitting individualized hemodynamic models while we're fitting the neural models, and these hemodynamic models are region specific. So on the left-hand side, I'm showing you the group average time to peak across the entire brain. And so, for instance, you see a slower time to peak in anterior PFC than in motor cortex. On the right-hand side, we're actually showing that these individual differences are actually relatively reliable. So the red boxes are showing the test-retest reliability of subjects, and the blue boxes are showing the degree to which subjects are similar to each other in two parameters that describe the HRF. This is just to give you an idea of what the weight matrices that we estimate look like projected onto the brain. So on the left-hand side, we have the symmetrized version, which we're not really showing the directions, and then the right-hand side, the arrows indicate the direction of the outward component. That is the component that's stronger outward than inward. And we have citations on the bottom for a bioRxiv paper that actually describes this method. We've done quite a bit of validation for it. I'm not going to go into each of the individual details, but if you're interested in things like how does this method respond to different choices of resting state preprocessing or how sensitive is it to motion, things of that nature, again, you can check out the bioRxiv papers. So once we build these resting state models, we're going to be trying to elucidate what the exogenous task forcing is. And we're going to use this using an approach termed activity flow, where we're going to be modeling the task activity as flowing through resting state architectures. So the main way that we do this is we look at the observations in task fMRI. We build predictions based upon the resting state model, and then we look at the difference between them. And that difference is going to be an estimate of the exogenous task effect. So the first step is that we estimate the latent neural activity using the deconvolutions that we built for each individual subject. Using a resting state model in the neural space, we make predictions one time step forward, and then we convolve these back to the bold time space, which gives us better control and better ability to compare the effects that we estimate with those that would be estimated using conventional techniques. Now, all of this just defines a filter, that is that it isn't requiring any knowledge of the task events. Once we have estimated this new time series, then we pass that onto our task fMRI software to perform GLMs. In our case, we're actually using AFNI, but any choice is applicable. So the application that we're going to look at to try to validate our method is looking at cognitive control tasks. And namely, we are looking at a data set called the dual mechanisms of cognitive control. Todd Braver actually has a poster that's going to describe this. And this data set contains 4 different cognitive control tasks. And subjects complete these tasks on 3 different sessions. These are repeated once each session. So 4 tasks times 3 sessions. And we also have resting state data. Each of these tasks has a high control demand versus low control demand condition. And it's that difference that's going to form the contrast of interest when we're comparing methods. So for each task, we're going to find the parcels that have a significant effect. That is, they have a significant increase in activity in the high control demand condition versus the low control demand condition. And using these parcels, we're going to compare both conventional techniques for TFMRI analyses and our proposed techniques using the MINDI modeling. So on the left-hand side, we have a histogram. The red bar is denoting the mean of significant parcels, and the gray dashes are showing the significance lines for p less than 0.05. And so we see that the red line is centered to the right of 0, indicating that we have, on average, an increase in statistical detection power. And then on the right-hand side, we have these parcels and their significance projected onto the brain for the pair t-test between the 2 different techniques. Again, for the AXCPT, same thing. Again, we see largely left lateralized PFC being one of the main players, and anterior insula, as we'd expect. The Stroop's a little bit of an outlier, so we also have tongue regions and superior temporal cortex, since it is actually a spoken Stroop task and scanner. And again, for the Q task listing, we're back to having a large effect in left lateralized prefrontal cortex and anterior insula, as well as some of medial prefrontal cortex. We looked at the omnibus test, that is taking all these tasks together and find the parcels that are consistently activated across all these different tasks. And then we looked between the 2 methods for these omnibus parcels. There are 34 of them. And we find that only one parcel actually doesn't have an increase in detection power due to our method. That's in the histogram on the left-hand side. And on the right-hand side, again, we have the projection onto the brain. Again, a large portion of activity is increased in the left lateralized prefrontal cortex, as well as bilateral anterior insula and some regions of left anterior medial prefrontal cortex. We also want to know whether this also increases our ability to detect the timing of neural events. Now, again, this is going to be moved back into the bold space, which we did for comparison with conventional analyses. But even though we are moving this into the bold space, we see a much more confined peak to the time series. So on the left-hand side, we have the time series of brain activity for the high versus low contrast for the AXCPT using our method, and the right-hand side using the original analyses. For both of them, in cyan, we have highlighted the 34 omnibus parcels. As you can see, around the 7th TR, that's when we expect the probe effect to be represented in the bold time series. We see a very pronounced peak using our technique, whereas with the original analyses, we see a much more slow and lagged effect that's drawn out over time. So we've actually increased the temporal specificity by which we can identify when neural events occur. So in conclusion, we've used MINDI to fit single subject resting state data to large scale neurodynamic models. These are nonlinear dynamical systems that are coupled with neurovascular mature models. And these models have directed connectivity, so we're able to identify the sign and the direction of interactions between regions. And we can do this very quickly. It takes 3 minutes to run per subject. Using these models, we've defined a filter to try to estimate what the latent task effects are. And by using this sort of filtering as a pre-processing step, that is, it doesn't require any modeling of task events when we actually do the filtering, we can increase the statistical power and the temporal precision of our TFMRI detections for cognitive control demands. So I'd like to thank my mentors and lab staff. Anxu Wang actually did a large portion of the TFMRI AFNI modeling, and our funding sources. If you have any questions, of course, contact me, and we have the code posted on GitHub.
Hi, my name is Takuya Ito. I am a 5th-year PhD student in neuroscience at Rutgers University, supervised by Michael Cole. Today, I'll be discussing a study where we looked at how differences in localized and distributed processes are revealed through the dissociation of task activations and task functional connectivity. All right, so in recent years, there's been an interest in hierarchical cortical organization. So in this one seminal paper by Feldman and Van Nessen in 1991, they showed that there is hierarchical organization as determined through anatomical wiring or anatomical connectivity, and this was estimated through track tracing techniques in monkeys. And more recently, using modern neuroimaging techniques through MRI, Josh Bird, John Murray, as well as Matt Glasser and Van Essen have shown that this myelin map is a good proxy for structural hierarchy. And so this can be obtained by just taking a contrast map of T1, T2 images in MRI scans. And then relatedly to relating structure to function, John Murray and Jiajing Wang had also shown that there is a hierarchy of intrinsic timescales, and this was actually estimated in non-human primates and monkeys using electrophysiology data. And so what we wanted to assess with this particular study was to look at hierarchical differences in localized and distributed function as estimated with task fMRI in humans. And so how we operationalized localized functionality was we used a task activation analysis across many different task conditions. And in particular, we focused on the task activation magnitudes. So this was the absolute value of those activations, average across many different conditions for each region separately. And so what we would find is that if there were regions that had strong responses or strong activation magnitudes across many different task conditions, we would infer that those regions reflected more localized processes. Now, to juxtapose localized processes with distributed processes, we use task state functional connectivity analysis. And so what we did here was we compared, for every region, we compared its average FC, or average functional connectivity, which is commonly referred to as weighted degree centrality in the network science literature, or global brain connectivity. And so what we did was compare the average FC from rest to task. And what we would infer is if the functional connectivity for each region would decrease during tasks, we would infer that that region was primarily involved in localized processes as well. All right, so the two main hypotheses that we came up with were the first, that we would observe differences in localized and distributed processes as determined by task activation and task functional connectivity analysis. And second, that these differences are reflected in hierarchical cortical organization. So we used the HCB dataset. All of our results that we showed here were replicated in an independent set of subjects in the HCB dataset. And we used all rest and all task state data. So what that included was the 7 tasks which spanned, or which contained in total 24 different task conditions. All right, so we first estimated the average task evoked activation magnitudes across these 24 task conditions. And again, we used the magnitude, so the absolute value of these activations. One thing to note is that we saw strong responses in the visual cortex, and we also were able to obtain an equivalent map of task FC changes for each brain area. And so what we found here was that it actually, or one thing to note here is that correlations tended to reduce during task states. And I don't want to get into the specific details of that particular observation, since that was covered in this 2019 bioRxiv paper, but I'm happy to answer questions about that as well. But we were able to correlate these two brain maps and found that overall task activations and task connectivity were negatively associated with each other. And so what we took away from that was that localized and distributed processes are negatively associated. Now, just because the brain activation map and the functional connectivity map were negatively associated, it doesn't necessarily imply that there's any relation to cortical hierarchy. And so what we did was we associated with the cortical hierarchy as determined by Daniel Margulies et al. In 2016, in which they were able to extract gradient or large-scale gradients of intrinsic cortical organization using resting state fMRI data. And so this is the first principal gradient visualized on the surface, or the principal gradient. And so we were able to correlate this map with our two task activation maps and our functional connectivity maps. And so what we found was that the task activation map, again, averaged across 24 conditions, was positively correlated with the principal gradient. And our task FC change map was negatively correlated with this principal gradient map. So the two takeaways here would be that areas with stronger task responses are lower on the cortical hierarchy, whereas areas with more FC reductions are also lower on the cortical hierarchy. I don't have time to go into the specific details of the following results, but I'd just like to mention them here, is that we were able to estimate different types of functional hierarchy. So this is the hierarchy of intrinsic timescales that we're able to estimate from resting state fMRI. So we were able to replicate John Murray's original 2014 study, which used electrophysiology data in monkeys, but also note that I know Dan Lurie had a similar abstract last year at OHBM. And we're also able to associate our task activation and functional connectivity change maps with the degree of regional myelin content, so myelin maps. And this was obtained from Josh Burt et al. 2018. And lastly, another way to think about localized versus distributed processes is that if a region is truly involved in distributed processes, that region's activity should be able to be predicted by the activity of other brain areas. And so we came up with this framework in 2016, in which we were able to predict the activation of a held out region as a function of other brain areas. So this activity flow mapping framework tests whether a target region's task activation can be predicted as a function of other brain regions' activity. In other words, a distributed process. So what you would do is if you wanted to predict the activation in this region J, you would take the task activations of all other regions in blue and compute the linear weighted sum weighted by the resting state FC to this purple region. And so in essence, what we found was that association regions were accurately predicted by activity flow mapping, whereas sensory motor regions were poorly predicted by activity flow mapping. And so this poor prediction likely reflects a more localized or an internal process that cannot be predicted by other brain areas. So just to summarize, we associate task-related activation and connectivity changes to hierarchical intrinsic properties, such as the principal gradient estimated from resting state fMRI, the timescale hierarchy, as well as more structural properties, such as the myelin mapping or myelin map. We use task activations and distributed task FC to infer or as proxies for localized and distributed processes, and that we found that these localized and distributed processes are negatively associated across the cortical hierarchy. Of course, there are several outstanding questions. The first is, are fMRI bold activations and FC changes good proxies for local and distributed processes? Would these findings or current task activation and task functional connectivity analysis, would these generalize to a wider set of tasks? And lastly, what is the actual causal structure of this hierarchical functional organization? In other words, what might the biophysical mechanisms be that are necessary and sufficient to explain the dichotomy of localized and distributed processes? And with that, thank you for listening. Feel free to email me with questions, and here is the citation of our preprint on bioRxiv. Thanks.
Hi, everyone. My name is Martin Rgaard, and I'm a researcher at the Neurobiology Research Unit, directly reporting to you from Copenhagen in Denmark. And I'm really excited to let you know about my work, which has the title, A High-Resolution In Vivo Atlas of the Human Brain's GABA-A Receptor System. So the GABA is the main inhibitory neurotransmitter in the human brain, and it has been implicated in several disorders such as epilepsy and autism. And one of the major components of the GABA system is the GABA A receptor located here on the postsynaptic neuron. So the GABA A receptor consists of 5 units, subunits into this pentameric structure. And in more than 50% of cases, it looks like this. So this is where you have the 2 alpha-1 subunits, you have 2 beta-2 subunits, and you have 1 gamma-2 subunits. But actually, there are 19 subunits that can go together and form the GABA A receptor. Now, what happens when GABA binds to this receptor is that it facilitates the opening of this channel. So chloride ions, which are negative, they can go through and make the inside of the neuron more negative, and thus inhibiting a response. In addition, you also have the benzodiazepine binding site located between alpha subunit and a gamma subunit. And as you know, when you take benzodiazepines, they have this sedative effect. The reason for that is that benzodiazepines are an agonist. So they also open the channel and facilitates this chloride flow. Now, if you've taken too many benzodiazepines and you want to stop this response, you want to take a different drug, for example, flumazenil, which is an antagonist. And when that binds to the benzodiazepine receptor, it blocks this channel and hence the response. Now, several different imaging techniques have been used to measure GABA throughout time. And if I'll just start with the in vivo techniques, MRI spectroscopy and positron emission tomography have been widely used. And what is really nice about MRI spectroscopy is that you're able to measure directly the GABA levels. So the pros are that it's quantitative, it's in vivo, there's no radiation as compared to PET. The problems are that often you define just a seed region and then measure GABA inside that region. And because you often have a large seed region, you will have a low resolution, and again, that will give you partial coverage as compared to, for example, PET. Now, PET imaging instead is where you have a radioligand. This is the case of flumazenil that you inject it into the body, it goes into the brain, and here it has a radioactive decay, and that is basically what you're measuring with a PET scanner. So the pros are that it's quantitative. You're basically measuring the uptake over time in a given region. And if you have a high resolution PET scanner, you can get 1 to 2 millimeters of resolution. Cons are it's radioactive, and the quality will be defined by the tracer and the PET scanner. Now, when it comes to ex vivo techniques, there's some important work that I need to mention. And the first one is the Allen Human Brain Atlas, which are used by thousands of researchers in many different fields. And this is really nice because this gives you the mRNA levels for many genes for each probe, so in many different areas in the brain. The cons are, again, it's ex vivo, it's fairly low resolution because you have distances between probes. And then finally, the units are in log2 intensity, so it does not directly reflect how much mRNA do you have available. Finally, we have a technique called autoradiography, and this is also where you use a tracer to get the quantitative concentration of GABA A receptors in picomolar per gram tissue. Pros are that it's quantitative, and cons are it's ex vivo, and usually it is low resolution. For example, this would just be a slice, but this does not give you like overall or throughout the entire brain. So the aim of this work is that we want to create a high resolution atlas of the benzodiazepine receptor, reflecting GABA A receptor levels. We have 2 requirements. First, it needs to be high resolution, and 2, it needs to be quantitative. Then after that, we want to validate it with mRNA, and we want to estimate the minimum mRNA level for translation into BCR protein. And how do we do that? Well, we want to use PET imaging where we get the high resolution using flumazenil, and we want to use ex vivo autoradiography with the tracer diazepam to get the quantitative in picomolar per grams of tissue. Then we can combine that into this high resolution atlas. And finally, we can validate our atlas using the mRNA levels from each subunit from the Allen Human Brain Atlas and see how each subunit expression, mRNA expression, maps onto the benzodiazepine density. So the first thing that we did was that we took 16 healthy controls from the SIMBA database, aged between 18 and 45. These were scanned in a high-resolution PET scanner, either 1, 2, or 3 times for 90 minutes using flumazenol. And for this, we used either a bolus or bolus infusion approach, and we also measured the radioactivity in the blood over time throughout the experiment. And this is just a simplified version of what it looks like. So we put the persons inside the scanner, we measured the activity over time, and the output of this is these images of total volumes of distribution, where we're basically measuring the total concentration inside a given voxel as compared to the concentration in plasma. And that gives us the total distribution volume. Now, of course, we did not use the ex vivo brains of these subjects that were scanned. So we had to go a little bit back in time to find this information. And actually, we had to go back to 1977 in a paper published in Nature by Brestow called High Densities of Benzodiazepine Receptors in Human Cortical Areas. And what's really nice about this is that they did autoradiography using this tracer to get specific binding throughout different regions. So you can see you have high specific binding in picomolar per gram protein in frontal regions, whereas for pons and other white matter regions, it's much lower. And as I said previously, we got the total distribution volumes from PET. Now we have the specific distribution volumes from autoradiography. So if we try to make the association between these two, we will have left the intercept, which is the non-specific binding that you got from the tracer. And when you do that for the different subjects and across scans, you get something that looks like this. So this is where you have the autoradiography data in association with the VT. You get a very nice correlation with just a small intercept here ranging between 0 and 1. So that means low specific binding. You can now aggregate that into this group association, again, where you have the BCR density from autoradiography and the BCR density from PET, and displayed here with a 95% confidence interval. So now, because you have this group association, you can now go back to the VT images, make a group average, and then convert that into densities. And then you get this. So here you see that there's a high receptor density in the cortex, especially in the occipital cortex. But if you look at the subcortical regions, it's low here in the brainstem and in subcortical regions. Now, this was our first validation. The second validation that we did was to compare our developed atlas with the mRNA expression. And here I'm comparing it to the widely used subunits, the alpha-1, the beta-2, and the gamma-2. And here again, you see a good correlation ranging between 0.6 and up to 0.87. It's more variable in the subcortical regions, and there can be many different explanations for that, but mainly that subcortical regions, they are both gray matter and white matter, for example, in the thalamus. Then you can also estimate the intercept, which is the minimum expression of mRNA that needs to be there in order to translate into BCR protein. You can also do a PCA of all the subunits and the BCR protein, and then you get a biplot that looks like this. So here you can see the subunits that are correlated with the BCR and the ones that are anti-correlated with the expression of BCR, which are the Rho1, gamma-1, and theta-2. Whereas these subunits here, they don't seem to have any correlation with the BCR. Now, finally, we can also make a rough parcellation of our density or of our atlas, and then dividing into these ranges. And the point that I want to make here is that if you want to use a parcellation that's structurally defined, I have to say that function is not necessarily always equal to structure. So, for example, if you take this region here and comparing it to here, you see that it ranges from above up to 1,000 and then down to below 800. So inside this region, the signal is not homogeneous, and you will also have a gradient pointing to this direction. So one should be careful with using regions of interest analyses, and ideally it would be nice to make a parcellation of the functional BCR density here. Now, to sum up, we have presented a high-resolution in vivo atlas of the benzodiazepine receptor. Everything is freely available at this webpage, where there's also data and code available. It provides a highly valuable tool for investigation of the GABA system and can be used as a reference for patient groups. And then finally, you can also combine this with other imaging data. That could be fMRI. It could be used to also other functional imaging where you want to evaluate the efficacy of pharmacological targets acting on the BCR. And finally, I just want to say thank you very much for your attention. I want to thank my collaborators. The paper is available here, so go have a look. And otherwise, just thank you for your attention. Thank you very much.